Welcome! Here we go. We're going to learn about cellular respiration for AP Bio. Okay, cellular respiration is the process by which you and other living organisms burn their food to get energy. We're going to look at this with our chemical reaction of glucose plus oxygen yields carbon dioxide plus water and gives off ATP as the cell's source of energy. So glucose is oxidized. Remember, oxidation is loss of electrons or hydrogen atoms. Oxygen is reduced. Reduction is gain of hydrogen atoms or electrons. In this process, 36 to 38 ATP are formed. That means that about 39% of the energy available in glucose is being taken out. It's about 39% efficient. If we look at a car, it only uses about 20 to 30% of the energy available in its fuel. So you are more efficient than a car. Just like in photosynthesis, we're going to have coenzymes of cellular respiration. These coenzymes have the same function. They are going to channel all the energy in those electrons to get them safely to their destination. Just like when we made the analogy of holding a child's hand in a busy parking lot. Your coenzymes in cellular respiration are NAD plus and FAD. Okay, NAD plus will accept 2 electrons and 1 hydrogen ion to form NADH. The reverse process is also possible. FAD accepts 2 electrons and 2 hydrogen ions to make FADH2. Just like we talked about different phases of photosynthesis, we'll use the same method in cellular respiration. This time there are 4 steps. The first step is glycolysis. Second step, preparatory or prep reactions. Third step, citric acid cycle, also known as the Krebs cycle. And the fourth step is the electron transport chain, similar to the electron transport chain in the sodium-potassium pump that you learned about. Here's a diagram of cellular respiration, just a big general overview. What I want to point out here is that glycolysis is going to occur in the cytoplasm of the cell, whereas the Krebs cycle and electron transport chain occur within the mitochondria, the powerhouse of our cell. Okay, first step, glycolysis. Glycolysis means split a sugar. Okay, that's exactly what's going to happen. Our glucose is going to split into 2 G3Ps. Okay, this happens outside the mitochondria in the cytoplasm. It does not require oxygen. We can break glycolysis down into 2 smaller steps. The first one is the energy investing steps. It's going to take 2 ATP to split that glucose into 2 G3P. And then from there, each G3P goes to the same process. The other part of glycolysis are the energy harvesting steps. What happens here is that G3P is oxidized by NAD plus to make an NADH. Okay. Then substrate level phosphorylation is going to occur. 4 high energy level phosphate groups are going to join with 4 ADPs to create 4 ATPs. 2 of these ATPs are going to go back into glycolysis to split the next glucose, which gives the cell a net gain of 2 ATP that it can use for its energy. During this process, the G3P is converted to a 3-carbon sugar, which we call pyruvate. Okay, as we go through this, we're going to make lists of inputs and outputs for each of our steps. So here's your list of inputs for glycolysis, a glucose, 2 NAD+, 2 ATP, 4 ADP, and 4 phosphate groups. Out of that, we will get 2 pyruvate, 2 NADH, 2 ADP, and 4 ATP total, 2 of which are a net gain for the cell. Okay, prep reactions. This is going to get everything ready for the citric acid cycle. It's going to occur in the matrix of the mitochondria. The matrix is the space, not the cristae. Okay, your pyruvate, that C3, 3-carbon molecule, is going to be converted to a 2-carbon molecule, which is an acetyl group. Okay, that acetyl group is going to attach to a coenzyme called CoA. What happens to that carbon that is lost? Okay, that gets converted into a carbon dioxide. This is one of the products in our cellular respiration equation. So in this process, pyruvate is oxidized, NAD+ is reduced. When the pyruvate splits into the 2-carbon compound and the 1-carbon compound, electrons are lost. These electrons are picked up by the NAD+, and that gives us the pyruvate being oxidized and the NAD+ being reduced. This again is going to occur twice per glucose molecule because we split that glucose molecule and we ended up with 2 pyruvate. And your chemical equation is at the bottom of the slide. Okay, here's our ins and outs for our prep reaction. We put in 1 pyruvate, 1 CoA, 1 NAD+. We get out 1 acetyl-CoA 2-carbon compound, 1 carbon dioxide, and 1 NADH. Again, we have to double this for every glucose because each glucose splits into 2 pyruvates. Step 3, the citric acid cycle. This also occurs in the matrix of the mitochondria. Okay, what's going to happen here is our 2-carbon acetyl group from the prep reaction that's being carried by the CoA is going to join with oxaloacetate, which is a 4-carbon molecule. 2 carbons plus 4 carbons gives us a 6-carbon molecule called citrate or citric acid. Okay, this citrate is going to get worked on by all of these coenzymes. Okay, the citrate is going to be oxidized. The coenzymes are going to be reduced. Okay, so NAD is reduced to NADH. That 6-carbon molecule becomes a 5-carbon molecule called ketoglutarate. Carbon dioxide is given off. And then NAD works on that 5-carbon molecule and turns it into a 4-carbon molecule. Our result is an NADH, a CO2, and an ATP. Then it's FAD's turn. It's going to convert the succinate into fumarate. FADH2 is given off. And last but not least, NAD+ is in there again, converting that 4 molecule back to oxaloacetate, and we get another NADH. Okay, this ATP that's made in the citric acid cycle also comes from substrate level phosphorylation. Again, this process has to turn twice for each glucose molecule because we'll have the 2 acetyl-CoAs to put in. Here's a picture of the citric acid cycle. We can start here with our pyruvate acetyl-CoA combining with the oxaloacetate to make citrate. Okay, then all of those substrates are oxidized, coenzymes are reduced, and we return to oxaloacetate, showing that the citric acid cycle is cyclic. Its reactants become its very own products, and it can continue on. Here are your inputs and outputs for the citric acid cycle. Input, an acetyl group, 3 NAD+, 1 FAD, 1 ADP, and 1 phosphate group. We will get out 2 carbon dioxide, 3 NADHs, 1 FADH2, 1 ATP. Again, double for each glucose. Before we go into the electron transport chain, I need to introduce you to cytochromes. Okay, cytochromes are proteins that carry electrons. One example of this is our iron-containing proteins in the heme group of hemoglobin. They can alter the rate between oxidation and reduction. With the heme group, the iron is reduced when it accepts electrons, and it's oxidized when it loses electrons. In your electron transport chain, there are several types of cytochromes that work in a series to allow that electron transport chain to occur. Okay, here we go. Last step, electron transport chain. Now, this is working in the cristae of the mitochondria, all those folds on that second membrane inside the mitochondria that increase surface area. What's going to happen is electrons are going to be passed from each of those cytochromes, carrier proteins, to the other, and redox reactions are going to occur. This is going to allow that hydrogen ion concentration to build up in the inner membrane space, which is going to allow for a concentration gradient, which allows work to be done. Oxygen is going to be the final electron acceptor here. It'll receive low energy electrons and combine with hydrogen to make water. Okay, this is our other product of our cellular respiration equation. What's happening here this time to make the ATP is oxidative phosphorylation. Okay, the hydrogen ions are going to go back into the membrane through the ATP synthase enzyme. As they do this, it's going to allow ADP to combine with a phosphate group to make ATP. From each one of those NAD molecules, NADH molecules that is used, we are going to get 3 ATP. When we use the FADH2 molecule, we only get 2 ATP. In your book, we need to make a correction. We'll talk about that the next time I see you. Okay, so this is the numbers give you the sequence of the protein complexes and the carriers that are in the cristae of the mitochondria that make that electron transport chain. You're going to have 3 protein complexes and 2 carriers that each have their own special function in this electron transport chain. A picture is worth 1,000 words. Okay, here I can point out your 1, 2, 3 protein complexes and your 1, 2 carriers. In the diagram you can see that the hydrogen ion concentration is being built up on the intermembrane space, and these will go through the ATP synthase to combine the phosphate group on the ADP to make lots and lots of ATP in the electron transport chain. The ratio of hydrogen ions in the inner membrane space compared to the outer membrane space is about a 10 times difference. 10 times more hydrogen ions on the inside as on the outside. Okay, so here are your ins and outs of your electron transport chain. Notice that there is a range here for the ATP that are made. We will talk about where that range comes from the next time I see you. In the electron transport chain is where the bulk of your ATP is being generated. This is going to differ a little bit for different types of cells. Okay, so I want you to take the little quiz that comes after this video, and I want you to fill in your graphic organizer that's in your notes for electron transport chain. Bring all of your questions to me and we'll get them answered, and then we'll continue on with what happens. How do we make ATP when we don't have any oxygen? Have a great day. I'll see you soon.